This video explains the pathogenesis of cancer. The mechanism of induction of tumor is termed carcinogenesis or oncogenesis or tumorigenesis. The agent which induce or the tumor-causing agent is called carcinogens. Pathogenesis of cancer is detailedly explained based on 4 theories. First one is molecular carcinogenesis, second is chemical carcinogenesis, third is physical carcinogenesis, and the fourth is biological carcinogenesis. First we will learn about molecular pathogenesis of cancer. It is also termed genetic mechanism of cancer. The basic concept is explained based on: first is monoclonality of tumors. Most cancer cells arise from a single clonal cell by mutation or genetic alteration of cells. For example, benign uterine tumors developed from single clone have either glucose 6-phosphate dehydrogenase A or B gene types, but normal myometrial cell has both A and B genotypes. Second, molecular theory is the field theory. Under the influence of etiologic agents, only few cells develop into cancer cells. This is called field effect, and the theory is field theory. Next is progression and multi-step process of cancer growth. Various etiological agent hit the cancer cell one after another, which result the cancer cell to grow further. This process is called multi-hit process. The cells formed shows the properties of malignancy like invasiveness and metastasis. Next, molecular pathogenesis is based on genetic theory. The cancer cells shows either genetic abnormality or normal gene with wrong gene expression. These abnormalities are due to inherited, that is, transformed from parents, or induced mutation by chemicals or viruses. This mutated cell transfer their character to next progeny or next generation or offspring and result in cancer. The final molecular pathogenesis is based on genetic regulators of normal and abnormal mitosis. There are 4 regulatory genes which controls normal cell growth. One is proto-oncogenes, it encode proliferation pathway, it promote growth. Second is anti-oncogenes, it inhibits the growth of genes. Third is apoptosis regulatory genes, it regulates cell death. And the fourth one is DNA repair genes, it repairs the damaged DNA which occurred during mitosis. It also controls the damage to the proto-oncogenes and anti-oncogenes. If there is abnormalities found in the above 4 regulatory genes, mutated cancer cells are produced. These are the failure that occurs to the 4 regulatory genes. First is activation of growth-promoting oncogenes. Then sometimes inactivation of anti-oncogenes, resulting to cell growth. Abnormal apoptosis regulation also happen, which may escape cell death. Finally, failure to repair DNA damage result in mutation. The major genetic properties, or the hallmark of cancer, are: number 1, excessive and autonomous growth by growth-promoting oncogenes. Number 2, refractoriness, that is, not sensitive to growth inhibition by growth-suppressing anti-oncogenes. 3 is escaped cell death by abnormal gene-regulating apoptosis. Number 4, avoid cellular aging by shortened length of telomeres and telomerase activity. Telomeres are DNA sequences and protein found at the end of chromosomes. This stabilize the chromosomes and also required for cell division. Fifth property is continued perfusion of cancer through newly formed blood vessels by the process cancer angiogenesis. Sixth is invasion and metastasis, that is, cancer spread to distant site. Seventh is failure to repair damaged DNA, which is found in mutated genes and cancer. Eighth is clonal aggressiveness, results to cancer progression and tumor heterogeneity with different characters. Ninth is cancer with sequential hit by multiple carcinogens. 10th is dysfunction of microRNA suppress oncogenes results in cancer growth. Example: oncomeres. The diagram illustrates the molecular basis of cancer. The normal cell is attacked by carcinogens, result in DNA damage, it escapes cell death, and form mutated cell. Then the failure of genetic regulators happens. The mutated cell transform to cancer cells. Then, the tumor progress into malignant tumor. The second is chemical carcinogenesis. The stages that happens in chemical carcinogenesis are initiation, promotion and progression of cancer. The first stage of chemical carcinogenesis is initiation, which is induced by initiator chemical carcinogens. The chemical agent may be single dose for short time or large dose for longer time. The change produced by chemical carcinogens may be sudden, irreversible, permanent. There are two types of chemical carcinogens which act as initiating agent. First is direct-acting carcinogens. Example, alkylating and acylating agent. This induce cell transformation without any metabolic action. Second is indirect-acting carcinogens or procarcinogens. Example, aromatic amines, azo-dyes. These chemicals undergo metabolism to become carcinogens. So it is termed indirect-acting carcinogens. Now let's discuss how the target cell is converted to initiator cell. First, metabolic activation take place. Indirect-acting carcinogen get metabolized in the liver by enzyme monooxygenase of cytochrome P450 present in endoplasmic reticulum. So, for activation of indirect carcinogens, the requirements are balance between activation and inactivation reaction of chemical carcinogen. Second is the gene involved in metabolic activation, that is, cytochrome P450 enzyme. After metabolic activation, the chemical carcinogens are reactive electrophilic. That is, now the indirect-acting chemical carcinogen is deficient in electrons. This reactive electrophiles binds to electron-rich portions of other molecules like DNA, RNA, and other proteins. Then is the target molecules. The reactive electrophiles targets the DNA, and it produces mutagenesis. The mutated DNA now act as initiated cells. Last step is how the cell turn into the initiated cell. The damage produced in DNA cells are permanent and fixed. The damaged cell is vulnerable to other carcinogen attack. After initiation, the next stage is promotion of carcinogenesis. Some examples of promoters of carcinogens are phenols, hormones, artificial sweeteners, drugs like phenobarbital. The properties of promoters are: they don't produce sudden change. It produce the action only after its application. The change produced is reversible. It don't damage the DNA, but enhance the effect of indirect-acting carcinogens. It promotes by further clonal proliferation and increases the initiated cells. After promotion, progression of carcinogenesis takes place. Once when the initiated cells rapidly increase in number, the cells shows the properties of cancer. This stage is termed progression. The diagram illustrates direct-acting carcinogens with no metabolic activation or indirect-acting carcinogens with metabolic activation attacks the target cell. The next phase is formation of reactive electrophiles which targets the DNA and cause permanent DNA damage. The damaged DNA is the initiated or mutated cell. This process is called initiation. Then, the mutated cell undergo clonal proliferation. This is the stage of promotion. The proliferated cells shows cancer cell properties. Then, it transforms to malignant tumor. This stage is called progression. Next, we will see how physical agents play the role in cancer pathogenesis. Physical agents are radiation and non-radiation induced. First, we will see about radiation carcinogenesis. Examples of radiation-induced carcinogens are ultraviolet light, ionizing radiation. Like chemical carcinogens, radiation carcinogens also undergo initiation, promotion, and progression stages. Let's see how ultraviolet light act as carcinogen. If UV light penetrates to skin for few millimeters from its sources, like sunlight, UV lamps, the damage is limited only up to epidermis, that is the outer layer of skin. Excessive exposure to UV rays cause cancers, like malignant melanoma. What happens when UV rays hit the cell? It sometimes induce mutation. It may also inhibit cell division, inactivate enzyme, also might cause cell death. The biochemical reaction of UV rays is it form pyrimidine dimer in DNA. If this DNA-damaged person exposed to excessive sunlight, the damage remain unrepaired. Patient with recessive hereditary disease, if exposed to UV radiation, also with defect in DNA repair system, are at high risk to develop cancer. For example, xeroderma pigmentosum is a recessive hereditary disease. When patient is exposed to sunlight, have risk to develop skin cancers. Next is about how ionizing radiations like alpha, beta, and gamma rays, X-rays, radioactive isotopes, protons, and neutrons cause cancer. The types of cancers caused by this radiations are leukemia, cancers of thyroid, skin, breast, ovary, lung, uterus, myeloma, salivary gland cancers. The occurrence of radiation dermatitis and subsequent malignant tumors of skin is found high in X-ray workers and radiotherapists without using safety measures. The mechanism by which radiation damages the DNA of the cell is the radiation directly alter the cellular DNA. Otherwise, the radiation take away the ions from water and other molecules of the cell. Then it forms highly reactive free radicals. This cause DNA damage. DNA damage produce mutagenesis. It also cause chromosomal breakage into two parts, which reattach to other chromosome, which is called translocation. When genome base pair is added, deleted, or changed, it is called point mutation. These all predispose to cancer. The effect depends upon the type, dose rate, frequency of radiation. It also depends upon the host factors like age, individual sensitivity, immune competence, hormonal influences, type of cell radiated. The second is carcinogenesis, caused other than radiation. Injury to tissues, such as stones in gallbladder, urinary tract, may predispose to cancer. Healed scars following burrs may increase the risk of carcinoma. Finally, we will learn how cancer is caused by biological agents. Parasitic, fungal, bacterial, viral infections may lead to cancer. For example, schistosoma hematobium, parasitic infection of urinary bladder, is associated with high incidence of squamous cell carcinoma. The fungus Aspergillus favus in stored grains liberates aflatoxin. When patient with hepatitis B virus infection consumes this aflatoxin, this leads to hepatocellular carcinoma. Prolonged Helicobacter pylori bacterial infection of peptic ulcer lead to gastric carcinoma. Let's detailedly look how viral carcinogenesis occur. Viral infections are transmitted by three ways. First is horizontal transmission. This virus pass from one human to other by direct contact. It is also transmitted by ingestion of contaminated water or food. It is also transmitted by inhaling the infected virus. The infection starts first at the outer surface of tissues, that is, the epithelial surface. Then it spreads deep into tissues. Then it spreads to other sites through hematogenous or lymphatic or neural route. Second way of transmission is parenteral route. It spread when the infected virus enters from animal or insects to human or from human to human through bites or needle pricks and enters the blood. Third way is vertical transmission. This infection is genetically transmitted, that is, from infected parents to children. Based upon the nucleic acid content, oncogenic virus are categorized into two types. First one is oncogenic virus with deoxyribonucleic acid, that is, with DNA. Second is oncogenic virus with ribonucleic acid, that is, RNA, or retrovirus. Both DNA and RNA oncogenic virus has 3 genes. First is GAG gene, this codes group antigen. Second is POL gene. This codes polymerase enzymes. The third is ENV gene, which codes envelope protein. Primary viral infection lasts only for few days or few weeks. It is cured by body's own or innate immunity or other immune responses. Persistent viral infection or latent infection will not obey to immune responses, so thereby produce immunosuppression, that is, the immune system does not works. Now let's see the mechanism of viral oncogenesis. The persistence of DNA or RNA oncogenic virus induce mutation in the target cell. This cause activation of growth-promoting pathway or suppress tumor-suppressing pathway. That is, activate proto-oncogenes and suppress anti-oncogenes. The cells of viral infected host enter cell cycle. This is transmitted to next generation. These oncogenic virus have the characteristic to grow by its own without any attack by other carcinogens. DNA viral oncogenesis may undergo replication or integration. Virus replicates or multiply in the host cell by lysis or breakage of infected cells and release virions. Then, the viral DNA integrate into host cell DNA. Replication brings cell death but no neoplastic transformation. Integration result in mutation and transform host cell into neoplastic. Let's see how RNA viral oncogenesis happen. Retrovirus has 2 identical strands of RNA, and the enzyme reverse transcriptase. Reverse transcriptase is RNA-dependent DNA synthetase which act as a template to synthesis a matching single-strand viral DNA. This single strand of viral DNA is copied by DNA-dependent DNA synthetase to form another double-stranded DNA or provirus. This provirus is integrated to the DNA of host cell genome and induce mutation and then transform into mutated cell. This retrovirus are capable to multiply. The host cell, which allow retrovirus to multiply, are called permissive cells. Viral replication begins when the provirus integrate into host cell genome. This result in the transcription or copied progenes, which then carries the information encoded in DNA to messenger RNA, finally forms components of virus particles. The GAG gene codes virion core protein. The POL genes codes reverse transcriptase. The ENV gene codes envelope glycoprotein. The above 3 components of virus particles are assembled at the plasma membrane of host cell. These virus particles released by budding off from the plasma membrane. Thus it complete the process of replication. Examples of DNA oncogenic viruses. Some papoviruses are human papillomavirus, which is associated with cervical cancer. Polyomaviruses associated with sarcoma in mice. Some herpes viruses, like Epstein-Barr virus, which is associated with Burkitt's lymphoma. Human herpes virus B, associated with Kaposi's sarcoma. Adenoviruses associated with sarcoma.in hamsters. Poxviruses in humans is associated with papilloma. Hepatitis B virus is associated hepatocellular carcinoma. Examples of RNA oncogenic viruses. Acute transforming virus, like leukemia sarcoma virus, in chickens, associated with leukemia and sarcoma. Slow transforming viruses, like mouse mammary tumor virus, in daughter mice, is associated with breast cancer. Human T-cell lymphotropic virus, HTLV1, in human, is associated with adult T-cell leukemia lymphoma. Hepatitis C virus, HCV, in human is associated with hepatocellular carcinoma. The figure illustrates the integration and replication of DNA and RNA virus in the host cell. First, the host cell is infected by DNA or RNA virus. The DNA virus enter into host nucleus. So the T antigen gets activated. Viral replication happen, and it forms virions. This newly formed virions are gathered inside the nucleus. Once the host cell burst, these virions are released. On the other hand, instead of replication, integration may occur in presence of active T antigen. Integrated genome form neoplastic cells. This is the mechanism of replication and integration when DNA virus enters host cell. Let's look what happen when RNA virus enter host cell. The RNA viral envelope fuses with plasma membrane of host cell. At the same time, viral genome and reverse transcriptase is released into cytosol. This reverse transcriptase synthesize single-strand matching DNA and then copy it to form double-stranded viral DNA, which is called provirus. This provirus integrate into host cell genome to formed, transformed, or neoplastic cell. Integration of provirus result in replication of viral components. This viral components are released by budding.